Hi, my name is Jennifer Zoe, and I'm a PhD student at UCLA in the labs of Jason Ernst and Eliezer Eskin. Today I'll be talking about identifying global patterns of epigenetic variation across individuals. Understanding molecular variation across individuals is fundamental to understanding variation in complex traits. Recently, molecular data has been collected across many individuals, which has allowed for a more comprehensive identification of variation. One common way to identify molecular variation is through QTL analysis. In this type of analysis, a molecular phenotype is collected in multiple individuals and correlated with a genetic variant. A similar approach is to do differential peak analysis. This analysis identifies regions that are variable between groups of individuals. While the previous two methods did separate analyses for each type of data, another approach is to do a joint analysis across multiple data types. One way to do this is through chromatin state analysis. In a paper from the Snyder Lab published in 2013, they used the ChromHMM software to identify combinatorial patterns of histone modifications in CTCF. They then identified variable chromatin states across individuals. The box on the red is a variable region across individuals, and the box on the right is a non-variable region. Although these approaches identify variable regions of the genome, none of these approaches identify whether specific patterns across individuals reoccur in the genome. They all look for variation locally in a specific consensus region. One reason that we might expect to observe reoccurring patterns of epigenetic variation across individuals is transregulation. Differential expression of trans factors can potentially affect the transcription of many other genes in the genome. The goals of this project are to, one, identify if these global patterns exist, and two, to see how they relate to other types of variation. To learn these recurring patterns across individuals, we propose to apply an alternate stacked version of the ChromHMM framework. In this framework, each mark from each individual is conditionally independent given the hidden state. In addition to allowing for joint analysis across individuals and marks, the HMM framework takes into account the spatial context of the data. While other methods rely on identifying a set of consensus regions across all individuals, the input to this method is the genome-wide histone modification signal. In this framework, each hidden state is a global pattern across both marks and individuals, and each cell of the emission matrix corresponds to the probability of observing a particular mark in a given individual, given the hidden state. We applied this framework to a previously published dataset of 75 individuals in the lymphoblastoid cell line. In this study, they collected 3 histone modifications that are associated with promoters and enhancers, and they used matched RNA-seq data for 54 individuals. Using the binarized data, we learned a joint HMM model across all individuals and marks. The emission matrix shown on the right is grouped by mark, and within each mark, the ordering of individuals is the same. Within each state, there are similarities in the emission parameters between the marks due to biological correlation between these promoter and enhancer-associated marks. This correlation was learned in our model, even though the model was agnostic to the mark labels. We computed the correlation between each pair of marks within each state, and we showed that the median correlation across states is high and that the median correlation is maximized using a 90-state model, which we used for subsequent analysis. While this analysis does not rule out confounding factors between samples, the consistency across different histone modifications suggests that the global patterns may be biological rather than technical. One application of this framework is to predict transregulators using the genome segmentation output by our framework. We predicted transregulators using 2 different criteria. First, we computed motif enrichments for the 90 states in the genome segmentation and required that the motifs were significantly enriched and had at least a log 2-fold enrichment of 1.5. Then we correlated the expression of each of these transcription factors and identified those that were significantly associated with the global patterns. Transcription factors that fall into both of these categories have a strong potential to act as transregulators, and we identify 18 genes meeting these criteria. Now we'll take a closer look at one of the predicted transregulators, REL, that I've highlighted in the heat map. REL is a subunit of NF-kappa B that mediates sequence-specific binding. NF-kappa B is a family of dimeric transcription factors that are known to act as transregulators in B cells, from which the lymphoblastoid cell line is derived. In addition to the REL motif being enriched in different regions of the genome, the REL protein is differentially expressed, and the expression levels correlate strongly with the state 37 in our model. Thus, the differential expression of REL can potentially affect many other genes. We also applied this framework to a set of a data set of autism cases and controls in the prefrontal cortex tissue. We trained models with numbers of hidden states ranging from 5 to 100, and we selected the 90-state model, which maximized the number of states associated with disease status after correcting for multiple testing of states and models. To more quantitatively visualize the associations of the emissions with the disease status, I plotted two box plots showing the diagnosis status on the x-axis and the emissions on the y-axis. We applied the same framework as the LCL dataset to identify predicted transregulators, and we identified 16 transcription factors matching these criteria. Moreover, 12 of these predicted transregulators are enriched in 2 of the states associated with disease status. Thanks for listening, and if you would like to hear more, please tune into my talk in the RegSys CAUSE on the 16th at noon. Thanks.